Hello everyone, welcome to KTHM e-learning center. Today we are going to learn about plant growth regulators which comes under subject plant physiology of third semester of second year MSc. So you have already studied about plant growth and development in your previous year, that is third year. So plant growth regulators, these are small chemical compounds which are present in the plant which regulates overall growth and development of the plant. So today we are going to learn about plant growth regulators and different types of plant growth regulators, also classes of plant growth regulators, major classes of plant growth regulators. So what we are going to learn today is role. We are going to study role of plant growth regulators in natural phenomena, also in artificial or agriculture. So what are plant growth regulators? Plant growth regulators are chemical substances which governs all the factors of development and growth within the plant. So same like human beings, growth of plants or development of plant is also governed or regulated by different chemical substances. Okay, and these chemical substances, these are called as plant growth regulators, phytohormones or plant hormones. These three terms are synonymous terms. Okay. So types of plant growth regulators. So naturally occurring plant growth regulators which are present inside the plant naturally. Even we have synthetic plant growth regulators which we make synthetically in laboratory. Okay. Uh, in this plant growth regulator, we have two types of plant growth regulators. First one which promotes the growth of plant, which is growth promoters, and second one which inhibits growth of plants, which are growth inhibitors. So when we have started with the growth, even in plant, that growth should be stopped somewhere at some point. Okay, so those functions are performed by growth inhibitors. So see, these are different types of plant growth regulators. So we are going to study major 5 classes today. Even there are more, but for today we are going to study 5 major classes of plant growth hormones or plant growth regulators. So plant hormones, these are having 3 types. Okay, growth promoters, growth inhibitors, and the hormones which are both. They have dual function of growth inhibiting and promoting as well. So in growth promoter, we have auxin, gibberellin and cytokinin. In growth inhibitor, which inhibits the growth, abscisic acid, which is also called as stress hormone. And ethylene is a gaseous hormone which is having dual function of growth inhibiting and also growth promoting. So these are 5 classes which are divided into these three types: growth promoters, growth inhibitors, and inhibiting as well as promoting in function. So see, these are 5 groups or 5 classes, major classes of plant growth regulators: auxin, gibberellin, cytokinin, abscisic acid, and ethylene. So we are going to study these 5 major classes today or 5 major groups today. So first is auxin. So auxin is the first hormone which was discovered in the plant, and it is discovered by Fritz Wendt. It naturally occurring auxin in plant is IAA, which is indole 3 acetic acid. Also, naphthalene acetic acid and 2,4-diphenoxyacetic acid are obtained obtained from synthetic source. So it is having brief use in agriculture. And precursor of auxin in plants is tryptophan. So tryptophan is the amino acid from which, so this is precursor in the plant by which through the one biosynthetic scheme, auxin is synthesized from the tryptophan, which is amino acid. So see how auxin was discovered, or how, even Darwin have worked on auxin. What he have worked? See, so Darwin in 1880 he have performed one experiment on coleoptile. Okay, so coleoptile is a region which it is a growth region which bends toward the sunlight. After formation of seedling, it bends toward the sunlight. So he have performed the experiment. So why? Because he was amazed that how these plants they bend. Plants lack motility completely. They don't have any motility or mobility. But still, this coleoptile bends toward the sunlight. So he was very keen. He has very keen interest to find out how this plant or how this coleoptile particularly bends toward the sunlight. So he have started performing experiments on it. So Darwin and his son, they both have performed experiments on this. What they have done is they have taken the intact seedling first, and they have observed that this intact seedling coleoptile bends toward the sunlight. Then what they did is they removed the tip of coleoptile. So the tip portion was removed. There was no curvature. So after excision of this particular tip, what happened is they put a opaque cap on it. Opaque cap which is not having any hormone or any chemical, just opaque simple cap on it, and there was no curvature. So that particular coleoptile, it didn't bend towards the sunlight. So by this particular experiment, we can assume, we can conclude that there is something chemical compound or growth at the tip region. Okay, why? Because when they remove the tip and when they keep the opaque block on it, there were no curvature. Okay, the coleoptile didn't bend towards the sunlight. So there is something at the tip. So after that, after few years in 1913, Boyson and Jensen, they have performed another experiment. What they have done is they have put a mica sheet, okay, towards the dark side of the coleoptile. So they have put the mica sheet towards the dark side of the coleoptile, and what happened is there was no curvature. It didn't bend at all. When second time, what they have done is they have inserted mica sheet on the light side. Okay, that time there was a curvature. Then what they did the next is they have removed the tip of that particular coleoptile and they put a gel block in between. So there is a gel block, and on that gel block, what they have done is, or gelatin block, they have put a coleoptile again. So didn't excise it at all. They didn't excise that particular tip completely. What they have done is first they have removed the tip, then they place a gel block or gelatin block over it, and then they have placed the tip, the removed tip over it, and it have shown again the normal growth which is bending. So this particular experiment, it shows that when mica sheet, so mica sheet is not permeable. Okay, so it is not water permeable. So when they put the mica sheet, there was no bending. But when they have put in when they put this particular gel block which was water permeable, gelatin which was water permeable, so it have bent towards the sunlight, or it shows the curvature. So when the particular thing which we put in between, or when we put it on the coleoptile, if it is water permeable, or that time only it shows the curvature. When that particular substance is not water permeable, it won't show any curvature. So next, in 1990, they have performed one experiment again. Pal, he have performed the experiment. So what he have done is he have removed the tip of the coleoptile again, and he have placed that coleoptile toward the one side. Okay, towards the one side of the region of that particular coleoptile, that tip, and even at that time it shows the curvature. So there was no change in curvature, or there was no change in bending towards the sunlight. So in next experiment performed by Wendt in 1926, what he have done is he have taken several coleoptiles, and he have taken all the tips, he have removed the tips, excised the tips, and he kept that particular tips of coleoptile on gelatin blocks. Okay, he have kept it on the gelatin blocks, and after few time, or after few hours, he have removed those tips from the gelatin. Okay, what he have done is he have taken a gelatin blocks. On each block, he have kept those coleoptile tips. After few hours, what he have done is that remove coleoptile tips and only gelatin block. He have taken only the gelatin top, as you can observe in this particular diagram. Experiment performed by Wendt. He kept that particular gel block on the coleoptile, removed coleoptile, excised one. Okay, and what happens is coleoptile bend. This particular thing proves that there was some chemical compound in that coleoptile. Okay, there was some chemical compound in that coleoptile which traveled to the gelatin block, and then gelatin block, it absorbs the chemical compound and it provided that chemical to the excised coleoptile, and then it bent towards the sunlight. So the chemical compound, or auxin, which was present in the coleoptile tip, it traveled to the gelatin block, and when we keep the gelatin block on excised coleoptile tip or decapitated coleoptile tip, it would grow to the, it would bend to the sunlight. So see, so how the coleoptile, it moves toward the sunlight. See, in the first diagram or in the first phase, as you can see, there is sunlight, okay, and there are these auxin molecules. So the pink one, those are auxin, okay. So this auxin, what happened is they have accumulated towards the darker side, okay. They have accumulated completely toward the darker side, and then they have bent to the opposite side toward the sunlight. So what happens with this auxin compounds, they accumulate towards the darker side so that plants would bend to the sunlight region, is towards the sunlight. So as you have seen here, the mica sheet, observe again. So mica sheet, it was inserted into the dark side. That is why it was unable to bend toward the sunlight, because the region where that auxin it accumulates, okay, that was blocked over there, right? Boysen-Jensen, observe the diagram again, the Boysen-Jensen one in 1913. See, they have put a mica sheet to the darker side. So at that particular region only, what we have, accumulation of auxin, and that is why it wouldn't bend. As you can see here, it haven't bent because at this particular side to the darker side, it was having what? The mica sheet. So what are functions of auxin? So first and most important function of auxin is cell elongation, or elongation of cells of stem and roots. It elongate the cell. It enlarges it by size. So important function is apical dominance. Okay, apical dominance, that means if you apply auxin on any shoot tip of a plant, it will grow upward only. It won't show axillary branches at all. What it will do, it will just grow upward, not at all axillary. So indole 3-acetic acid, IAA, in apical bud, apex of the tip, suppresses the growth of lateral buds. That is, plant would grow only at the upwards manner, only upward, and not by the lateral bud. So there won't be a branching, lateral branching like this. Plant will grow into the straight direction. Second one is induces parthenocarpy. Parthenocarpy, that means the development of fruit without fertilization, right? So what it will do, so this parthenocarpy, this particular function is very important in making seedless fruits like grapes and bananas. So why they are not having seeds? Because of the parthenocarpy, they have developed the fruit without fertilization. So it prevents premature fall of leaves when we apply auxin. So there are sometimes, because of the nutrient deficiency or the water scarcity, premature leaf fall is there. So leaf, when they are green in color, they starts falling. So naturally what happens is leaf turns yellow and then they start falling. But sometimes due to drought or due to any nutrient unavailability, what happens is the leaf starts falling when they are green in color. So that time if you apply auxin, it will prevent the premature fall of leaf, flowers and fruits also. So it is also useful in stem cutting and grafting. So stem cutting and grafting, these two practices, these are very important in pomoculture and horticulture. So when we grow fruits and flowers, it is very important to have a good grafting and cutting procedure. So what we do in cutting and grafting is we make exact clone of that particular fruit tree or flower tree. So for example, there is one fruit tree, for example mango, it is giving very good yield, number of fruits are very good, pulp quality is very good, sugar content is good, pulp color and aroma is also good. So what we need to do is we need to make a clone. So that time we won't require to have a seed and then produce a plant. What we can do is we can vegetatively propagate it. That is the easiest method. By that particular method you can even produce exact clone of plant, and it is not time consuming. So if you use the seed, in the seed there is variation, okay, it is not exactly same as this parent plant. So that time what we do is, in the horticulture and pomoculture practices, we use the grafting and stem cutting method. So that when we cut the stem for vegetative propagation, or when we graft the two CN and stalk for the vegetative propagation, we require the hormone, we require artificial hormone which will initiate the rooting and shooting. Okay, so that time we can apply artificial auxin to it so that it would initiate the rooting, and then rooted plant we can keep it into the soil and we can grow the new plant. So it promotes flowering in few plants, for example pineapple, is not in all plants. So 2,4-D is a auxin which is used as herbicide. So herbicide, it is to kill weeds, unwanted plants which are present along the crops. They are killed by this 2,4-D, which is also auxin. It kills plant But and the specialty is when we have a monocot crop, okay, so for example like maize, wheat, jowar, bajri, but that time we have the weeds, dicot weeds will present inside it. That time it is very useful. We can spray 2,4-D, which is Auxin, and we can kill all these weeds by using this artificial Auxin, and it doesn't affect monocot at all. Also, Auxin is having smaller role in cell division and xylem differentiation. So next important class is gibberellin. So there are more than hundreds of gibberellin present. Okay, they're acidic in nature. These are found in higher plants and as well as fungi. So how these are discovered? There is one very interesting story behind it. In Japan, it has been discovered. So in Japan, they have a disease on the rice plant, which is called as mad seedling disease or foolish seedling disease, which is Bakane disease. Okay, Bakane disease, that means mad seedling or foolish seedling. What was the symptom of this disease? Those rice plant, they used to grow taller and taller, very high height, very huge. Okay, so because of this tallness of that rice seedlings, they used to call it as a mad seedling disease, or this larger seedling. So when they observed it, Japanese scientists, there was a fungal infection on that particular rice plant. So fungus was Gibberella fujikuroi. Okay, and from this fungus, they have isolated gibberellin, which is a plant hormone and which is also present naturally inside the plant. See, this is the foolish seedling disease. So in A, you can see these seedlings which have grown in height unnecessarily. That is why we call it as a foolish seedling disease or Bakane disease. So what are functions of gibberellin? So most important function is induces flowering. Okay, so when gibberellin is there, it is activated by sunlight. Okay, it is activated by sunlight. It goes to the tip or the bud region or primordia region where it induces the flowering. It promotes elongation between the nodes. Okay, it promotes elongation between the nodes just before flowering in the plants like cabbage or beet. It delays the senescence. Senescence is naturally occurring phenomena in which the old aged plant parts, they are fallen from the plant. Okay, this is called as senescence. So aged leaf, root, stem, these are detached from the plant naturally. The gibberellin having function to delay it. When we want to delay the senescence or the leaf fall, we can spray gibberellin so that it would stop the senescence, or it would delay the senescence, not stop, it would delay the senescence. It induces parthenocarpy. Again, to form the seedless varieties, we can use gibberellins. Parthenocarpy is formation of fruit without fertilization. So elongation of the stem is the function. As you can observe in the Bakane disease, there was elongation. So elongation of stem, and it reverses dwarfism. So if we want our plant to grow taller than natural, we can use gibberellin. Or if we want our plant, for example rice plant, to grow dwarfer, or in short, what we can do is we can use few gibberellin we can use few inhibitors which inhibits the gibberellin, and that is why we can have the dwarf plant. So there is no gibberellin in the plant, that is why they won't grow in the height, and we don't have the unnecessary elongation of the stem. So it induces the maleness, that is it induces formation of anthers in certain plants like cannabis. It induces formation of hydrolytic enzymes. For example, lipase, amylase in endosperm of germinating grains and barley seeds. So what these enzymes which breaks down complex molecules to the simpler molecules. So this is having function in seed germination. So gibberellin is also having function in seed germination. So when seed is in dormant condition, okay, from at the time of germination, the gibberellin is active. It induces the germination by, so in the seed, all the food is storage in form of complex molecules, for example starch or protein. So what at the time of germination, what we need to do is we need to convert it into complex form to simpler form, for example monosaccharides and amino acid. So at that particular time, what we require is we need the hydrolytic enzyme. So this hydrolytic enzyme, for example lipase, amylase, these are activated by the gibberellin. So if we want to make our seed germinate, or from we want to break the dormancy, what we can do is we can use gibberellin so that it would change the seed from dormant period to the germinating seed. It breaks dormancy, that is the next function which we have already discussed. Cytokinin. So as the name suggests, cytocell, it is having very important role in the cell cycle or cytokinesis process. So it is naturally synthesized in plants at the time of cell division. When we apply cytokinin, cell division occurs rapidly. It is present in the root apices, shoot, bud, young fruits, wherever or anything, anywhere where we have cell division or growing, that in that particular area cytokinin is present. So movement of cytokinin is basipital and polar. Naturally occurring cytokinin, these are zeatin. It is present in coconut milk, and isopentyl adenine. Synthetic are kinetin, benzyladenine, diphenylurea, and Philip White is a person which have discovered cytokinin, zeatin in coconut milk. So functions of cytokinin, it promotes lateral and adventitious shoot growth. So as you can observe here, auxin is having function of apical dominance. When we apply auxin, plant will grow in upward direction, it will show apical dominance. But when we have cytokinin, when we spray cytokinin or plants, it will promote the lateral growth and it will promote the adventitious shoot. So cytokinin is having antagonistic function to the auxin. They are having opposite function to each other. So it helps in overcoming apical dominance. So if particular plant shows increased height, or if that plant is having apical dominance, it is only increasing in height and not showing any axillary branches or axillary shoots. So what advantageous shoot? What we are going to do is, if we spray cytokinin, if you apply cytokinin, it will show formation of axillary branches too. So at the time of germination, it promotes expansion of dicot leaf. Okay, and important function is it stimulates formation of chloroplast in leaf. So formation of chloroplast, in that particular process, cytokinin is involved. So it promotes nutrient mobilization. Nutrient mobilization as in from leaf to other parts of plant. It promotes the nutrient mobilization so that nutrients are sent over to other parts of plant. Interesting function of cytokinin is literally delete the leaf senescence. So there is one experiment performed, that if we take a green leaf, complete green leaf, and if we apply cytokinin at particular area in the patches, okay, so if I have the green leaf, I would apply cytokinin in the patches. So after few days, okay, the whole other leaf, it would remain green, it would remain yellow, it would become yellow, but patches at we have applied the cytokinin, it would remain the green in color, it would remain green in color. Why so? Because we have applied cytokinin and it would delay the senescence, and this is called as green island or island of the cytokinin, which have remained green even after other parts or other leaf is become yellow. So next interesting enzyme is abscisic acid. Abscisic acid is also called as stress hormone. So literally almost all the stresses which plant faces, like drought, flood, these can be tackled by abscisic acid. So this is the important hormone. This is called as growth inhibiting hormone. Because of this stresses, abscisic acid stops or inhibits the growth of plant for a while. That is why we call it as a growth inhibiting hormone. And it acts as an antagonistic to gibberellic acid. Okay, abscisic acid is against the gibberellic acid or gibberellin. So when we need to stop gibberellin, or when we need to stop function which are performed by gibberellin, what we can do is we can apply abscisic acid so that it would immediately suppress the gibberellic acid amount in the plant, and it inhibits the all functions which are regulated by the gibberellic acid. So it inhibits plant metabolism and regulate abscission. Abscission, as it removes the plant. It is involved in leaf fall, fruit fall. Okay, so abscisic acid and ethylene, they are together involved in this particular. So it increases tolerance of plant. So tolerance against the several diseases and abiotic stresses also. So what are function? It induces abscission of leaves and fruit. Why it does so? So when there is drought, okay, or when there is water scarcity, when water is not available, proper amount of water is not available, so there is no point of growth. Okay, even if you grow leaves, okay, even if you grow flowers and fruit, there is no water, ultimately the plant is going to die by consequence. But so first important thing is survival. So what is important than reproduction is survival. So what abscisic acid do is it induces abscission of leaf, and it overall stops the formation of leaf and fruit. So after abscission of leaf, obviously there are no fruit formation, so it uh, abscise the leaf, or it promote the leaf fall, so that there is less water loss. So what happens is leaves are having stomata, so through this stomata there is transpiration, that water vapors comes out of this stomata. So to prevent this loss of water through the stomata, or to stop the photosynthesis, even photosynthesis requires water, right? So to stop this particular excess use of water, or to prevent loss of water, what it does, it removes the leaf, fruit, or unnecessary plant. So first agenda is to survive. It inhibits seed germination. So as we have already discussed, gibberellin is having, or gibberellic acid is having antagonistic function to abscisic acid. So dehydration of fruit. When embryo is formed, the cell is hydrated. Embryo cells are hydrated. But from embryo to seed formation, or dehydrated seed formation, abscisic acid works. It makes the seed completely dehydrated, 90% dehydrated. Okay, when you observe any seed, it is dry, right? Why? So that it won't germinate, it would have its dormancy period. So abscisic acid keeps the seed dehydrated, and it stops the seed germination, it inhibits seed germination. But when there is availability of water, gibberellin is activated, and it acts against the abscisic acid, and it would promote uh, enzymes like amylase and lipase, and there would be germination. It accelerates dormancy in seed. So why it is important? Why somebody wants seed to not germinate? See, when we store the grains like wheat, jawar, bajri, or rice, when we store this grain, we don't want them to germinate. If they are germinated, they are wasted. So if we want to keep the grain for more time period without germinating, what we can do is we can we can apply abscisic acid to it, so that it won't show any germination. That is why they are having longer time of the storage. So another next important function is it stimulates closure of stomata so as to prevent transpiration due to water stress. So what happens is water loss is there in the transpiration process through stomata. So this opening and closure of stomata are having guard cells. When transpiration takes place, the guard cells, these are open, and water vapors they goes out. Okay, so to stop this particular water loss, when there is water scarcity or drought, what abscisic acid does is it closes the stomatal aperture. It closes the stomatal aperture so as to prevent loss of water to abscisic acid. So ethylene, this is a fifth class of enzyme. Okay, and this is the only hormone in the plant which is gaseous in nature. So this particular hormone is gaseous in nature. Also, it is very useful in artificial form. So it acts as an growth promoter as well as an inhibitor. Ethylene is promoter also and inhibitor also. It occurs in gaseous form. Okay, it is synthesized in ripening fruit. So when the fruits are premature or not mature, ethylene induces maturity in the fruit, and it ripens the fruit, and then leaf fall is there. So ethylene is having role in ripening of fruits and the tissues undergoing senescence. It promotes the senescence. It promotes the aging of the plant. It regulates the many physiological processes, and it is most widely useful hormone in agriculture. So what are functions of ethylene? So it promotes ripening of fruit, as we have already discussed. Even artificially, it has been used. Most popular in banana. Many people, many horticulturists or many permaculture permaculturists, what they do is they use ethylene, or any fruit vendors. Even fruit vendors, they use ethylene so as to promote ripening of banana. Within a day, banana turns yellow from green to yellow if we apply the gaseous form of ethylene. So it promotes ripening of fruit. It controls epinacity of leaf due to the low light, or due to the low light availability. There is epinacity of leaf. It also controls it. Okay, so when there is low light, ethylene would show leaf curvature. Okay, so that is called as epinastry of leaf. It breaks the seed dormancy. Okay, as it promotes the ripening or senescence or any kind of what we can say. Just a minute. It breaks seed dormancy again. That is, it is having function against the abscisic acid, and it is having functions along with the gibberellin. So gibberellin and ethylene, they both breaks the seed dormancy, but ethylene is not having that much important role in breaking of seed dormancy. It is having most important role in fruit ripening, leaf abscission, or leaf fall, fruit fall, flower fall. But gibberellic acid is having upper hand in the breaking of seed dormancy. It stimulates rapid elongation of petioles and internodes. So when there is excess water or flooded condition is there, that time if the plants grows in height, so that to avoid the damage because of the excess water, so that time ethylene show its function. It increases it elongates the petiole and internode so as to prevent from the excess water decaying due to excess water. It promotes senescence and also abscission of leaves and flowers. So this is the important function of ethylene in leaf fall, in fruit fall, and in flower fall, as it promotes senescence and abscission of leaf and flowers. Again, important function, it induces the root growth. Okay, so root growth which grow in size, which dig deeper into the soil so as to find out the water. So that particular root growth is induced by ethylene. So the root will go deeper and deeper to find water. Even root hair formation is induced by ethylene. So absorption surface increases. So when there is water scarcity, absorption surface will increase so that it would absorb water. So again in here it is having antagonistic function to abscisic acid. So in drought condition or in flood in drought condition or in water state, what abscisic acid, the stress hormone, does it's it would increase the root in size. Okay, the tip, it will elongate the root, but it will stop root hair formation, right? So this ethylene, it induces both root in height also. It will root will dig deeper to the soil so as to find out water, and even root hair formation is there so as to increase absorption surface. Also it induces apical hook formation in dicot seedlings. The dicot seedling, when there is scarcity, when sunlight is not available, there is formation of apical hook. It is induced by ethylene, and it can be tackled by using auxin, because auxin is also antagonistic to ethylene. So this was these were five classes of enzymes, or five classes of plant hormones, not enzymes. These are five classes of plant hormones. First one is auxin, gibberellin or gibberellic acid, cytokinin, abscisic acid, and ethylene. These are five major classes. Even we have racinosteroids also. We have few secondary metabolites which act as phytohormone or which induces few growth and development phenomena in the plant. But these are basic five major classes. These are having different function. These enzymes, these are present in very smaller amount. These hormones are present in very smaller amount, but they show a very good effect on plant growth and development. In general, we can say the growth of plant or development of plant is completely governed by the plant hormone or phytohormones. I hope you have understood these five classes of phytohormones, plant growth regulators or plant hormone. Mostly they are called as plant growth regulators. So assignment for you is write in brief about discovery of auxin, or how many experiments they have performed. Explain those experiments in short, and write a few words about auxin also. And you can mail it on my mail ID shweta@kthmcollege.ac.in. So what was the summary of today's session? We have studied about concept of the growth regulator, right? So how these growth regulators, they govern functions in plant, how they govern functions in metabolism in plant, how they govern the development and growth of plant. We have studied five major groups of plant growth regulator. Even there are more, but these are considered as five major classes or five major groups of plant growth regulator. So in next lecture, we are going to study about physiological role of auxin and cytokinin in detail and vice versa. I hope you have understood these classes very well. Thank you so much.